Hello boys, welcome back to another interesting session of video tutorials, and this session is meant for class 11th students, class 11th biology students, and this is for 11th September 2020. Our memory helps us in tracing out what we have been discussing in our previous sessions and linking that knowledge with the present knowledge so that we have a clear understanding of the concept. Yesterday's session we were talking about the structure of human excretory system as well as the structure of nephron, and we said nephron is the unit of excretion. So don't get confused with neuron. Neuron is a narrow cell. Nephron is the cell present inside the kidney and it helps in excretion. So one of the forms that human beings excrete out all the metabolite waste is through urine formation. What exactly happens and how it happens? It includes urine formation involves three main steps. First, glomerular filtration. This is number one, or it is also called ultrafiltration. Second, tubular reabsorption, second step. And third step is tubular secretion. So step number one, ultrafiltration. Step number two, tubular reabsorption. Step number three, tubular secretion. So let us discuss one by one. First we go to glomerular filtration. Glomerular filtration. The first step in urine formation is the filtration of blood, which is carried out by glomerulus. Remember that glomerulus, network of arteries and veins, and that glomerulus is present in the Bowman's capsule. So it's a network of arterioles, and this process of filtration of blood that takes place in glomerulus is called glomerular filtration. On an average, 1100 to 1200 ml of blood is filtered by the kidneys per minute, which constitute roughly 1/5 of the blood pumped out by each ventricle of a heart in a minute. Now, the glomerular capillary blood pressure causes filtration of blood through three layers. So there is blood pressure, internal pressure. Endothelium, first one, endothelium of the glomerular blood vessels, epithelium of the Bowman's capsule, and the basement membrane. Between these two layers, epithelial cells of Bowman's capsule, remember podocytes, yesterday I said, and are arranged in an intricate manner so as to leave some minute spaces called filtration slits or slit pores. Blood is filtered so finely through these membranes that almost all the constituents of the plasma except the proteins, now see why proteins except the protein, because of the size, pass on to the lumen of the Bowman's capsule. Therefore, it is considered as a process of ultrafiltration. So glomerular filtration or ultrafiltration. And what is so great about this? The amount of the filtrate formed by the kidneys per minute is called GFR. Remember this? I will repeat. The amount of the filtrate formed by the kidneys per minute is called GFR, glomerular filtration rate. GFR in a healthy individual is approximately 125 ml per minute. And many times whenever persons are sick, blood test will be done. Along with the blood test, GFR also will be done. And what about the amount? 180 liters per day. My God, that is the importance of kidney. So in a healthy individual, the kidneys will be doing filtration of 180 liters per day. That is the greatness of the kidneys. So basically ultrafiltration occurs because the pressure of the blood flowing in the glomerular capillaries is higher than the pressure of the filtrate in Bowman's capsule. It does not require the expenditure of energy by kidney cells. So it is not active, passive. The quantity of glomerular filtrate formed each minute in all the nephrons of both the kidneys is called GFR. In a normal person, as I said just now, GFR is about 125 ml per minute. So this is what the greatness of glomerular filtration. Look at this. So afferent arteriole, the blood is coming inside and it's entering inside, and you got this is the Bowman's capsule. We have got podocytes, the epithelial cells. Then of course there's a lumen, Bowman's space, and then there is mesangial cells. And once the filtrate comes inside along with the blood, glomerular capillaries will do the work, and finally it goes through afferent arteriole. So the direction is in, go through the process of ultrafiltration and out. This is what happens in the glomerulus. And then to be specific here, you can see afferent arteriole with the high blood pressure, high pressure within, enters inside and then go through the glomerulus, the network, and then the filtrate takes place. So urine is coming out, and once the blood having the urine goes off with useful materials, there is exchange in the filtrate and goes through afferent arteriole and back. And all this is happening in the cup-shaped structure, and that cup-shaped structure is called Bowman's capsule. And the urine that is filtered out, comes down from glomerulus to PCT, proximal convoluted tubule. And how exactly it happens? Now, what are all the materials that are taken back to the body? Let us discuss. Now, look at this. So, what we were talking about ultrafiltration is this. Now, we cannot afford to lose, our body cannot afford to lose some of the nutrients such as vitamins, proteins, amino acids, hormones. So they have to be reabsorbed. And then similarly, the tonicity should be maintained in our body. For that, water is required. Remember, in junior classes you studied 3 different solutions, right? One is called hypotonic solution, another is hypertonic solution, and third one is isotonic solution. So these, for your memory sake, I will just make you recollect what is hypertonic solution. Hypertonic solution, I will say, is concentrated solution. When I say concentrated solution, in your chemistry lab, biology lab, you have seen on the bottle it is written conc HCl, concentrated HCl. What do you mean by that? In that, water portion is less and HCl portion is more. That means hypertonic solution is a concentrated solution. Means any concentrated solution contains higher solutes, number of solid particles are more. And when solid particles are more, what is less? Naturally, water is less. So solvent is less. So that is hypertonic solution. And what about hypotonic solution? It is a solution that contains reverse, low number of solutes and high number of water molecules. That means hypotonic will be dilute solution. So I want you to remember that hypertonic solution is conc solution. Hypotonic solution is dilute solution. Also, you can remember it as hyper means lower. What is lower? We will take water. Hyper is lower. What is lower? Water lower. Hypo is high. What is high? Water. So if you take only water as the base, you can say hypertonic, hyper lower. That's how you can remember. And if hyper is lower water, hypo will be automatically higher water. That's how you can remember. So keep this in your mind, because we'll be often talking the solutions at various levels as it processes through these words like hypertonic, hypotonic, isotonic, like that. And what about isotonic? In both the ways, in both the solutions, both left and right, the number of solvents and solutes is equal to number of solutes and solvents on the other side. So both the solutions will be having similar or same number of solutes as well as solvent particles. Then it becomes isotonic. So from glomerulus here, now the filtrate starts into coming to PCT, proximal convoluted tubule. So in proximal convoluted tubule, the reabsorption of materials start. That how we are calling second step as tubular reabsorption. It is the second process in the formation of urine from filtrate. As much as 99% of the material in the filtrate is reabsorbed. That is the greatness. Preventing the loss of water, that is the aim. You can see water is getting reabsorbed, nutrients are getting reabsorbed, ions are getting reabsorbed, particularly Na plus, Cl minus, HCO3 minus, K plus, and in that particularly there is sodium potassium pump in our body, which should be always balanced. Then only person will be healthy. Na plus plays crucial role in functioning of heart, and K plus as well. So the K plus and Na plus balance has to be maintained in our body. And then as a consequence, urine contains mostly waste materials and excess water. So only waste materials will be there in the urine. About 65% of the glomerular filtrate is normally reabsorbed in the PCT, proximal convoluted tubule. Before reaching to loop of Henle, this U-shaped structure, this is called loop of Henle. All of you know this U-shaped structure. And this Henle's loop will be having one tube which is going down. This is called descending limb. This is descending limb. And then there is a U-turn. After U-turn, the limb is going up. This is called ascending limb. So you got a descending limb and a turn and then ascending limb. So before it reaches the loop of Henle, what is that happening here in the descending loop? Glucose, amino acids, vitamins, hormones, sodium, potassium, chlorides, phosphates, bicarbonates, much of water and some urea from the filtrate is absorbed. Sulfates and creatinine are not reabsorbed because they have to be thrown out. Sodium and potassium are reabsorbed by primary active transport. Active means we are spending energy. Glucose and amino acids are reabsorbed by secondary active transport. And water is reabsorbed through osmosis. Chloride ions, urea, and other solutes are reabsorbed by diffusion. So the filtrate is now becoming isotonic to blood plasma in Henle's loop. And what about in descending limb of loop Henle? Water is reabsorbed due to increasing osmolality of interstitial fluid. Sodium and other solutes are not reabsorbed here in the descending limb. Sodium and other solutes are not reabsorbed. The filtrate becomes hypertonic. You see here it is isotonic. In the descending lobe it is becoming hypertonic to blood plasma. Ascending limb of Henle loop is impermeable water. It is impermeable to water, ascending, but permeable to K plus and Na plus and Cl. Look at this. So, and it is partially permeable to urea. So the filtrate becomes hypotonic. I will write here it is hypertonic here. Whereas in the ascending limb of Henle's loop, it is becoming hypotonic. The filtrate is becoming hypotonic to blood plasma. And once it reaches here, this is called DCT, distal convoluted tubule, this portion. So in distal convoluted tubule, there is active reabsorption of sodium, as you are seeing. Active reabsorption of sodium ions from the filtrate under the influence of, now you must remember, aldosterone. This is very, very important. This plays crucial role. Aldosterone will make sure that sodium ions are reabsorbed. And water is reabsorbed here, as you are seeing. Water also reabsorbed here under the influence of, again, another hormone is there, ADH, anti-diuretic hormone. And these are both aldosterone and ADH are secreted by pituitary gland. Aldosterone anterior lobe and ADH posterior lobe of pituitary gland. This makes the filtrate isotonic. Again, once it comes to DCT, it becomes isotonic to blood plasma. This is what you must remember. And further, when it comes to CD, collecting duct, what's happening? Further reabsorption of water takes place and the filtrate becomes hypertonic. Hypertonic. Once it becomes hypertonic to blood plasma, sodium is reabsorbed in the collecting duct under the influence of aldosterone. The filtrate is now called urine. This is how the urine is formed. Urine formation takes place. And the urine is hypertonic to blood plasma. And tubular secretion, last one. It refers to the addition of selected materials from the blood to the ultrafiltrate and thus increasing its volume. And it occurs mainly by active transport. It takes place in DCT and PCT. This is how the nephrons play crucial role in formation of urine. And then once this is formed, all the collecting duct, all the nephrons will put the urine in the collecting duct. Collecting duct comes and form of ureter, and both the ureters left and right will join to urinary bladder, and from urinary bladder the urine has to be extracted. So the expulsion of urine from the urinary bladder is called micturition. Again, this is one more question. MICTURITION, micturition. What is micturition? The process of expulsion of urine from urinary bladder, and it is a reflex process, and it is normally in grown-up children and adults. It can be controlled voluntarily to some extent, but in some people this will not be there. The reflex process, there will be a problem, and urine may not be expelled from urinary bladder. So that causes a lot of problem to them. And then the kidney specialist has to take care of whether there is any disturbance in aldosterone, whether there is any disturbance in ADH, or in the making of the different pressures, like I will show you some here, like this. So it is osmolality. So the pressures are to be maintained in this way, 300 to 400, then 600, you can see here. And vasa recta. In vasa recta, this is to be maintained, the pressures, and various materials reabsorption you can see, and in the whole nephron you can see how the pressure is to be maintained, osmolality is to be maintained, then only the formation of urine takes place. So please draw the diagrams, try to understand what I have explained.